have you ever really thought about what's happening inside your body when you get sick? I mean, it's a microscopic war zone. Today, we're going to go behind the front lines and look at the brilliant strategies of our greatest weapon in that war, antibiotics. Let's find out how these smart drugs hunt down their targets.
It's a question that sounds so simple, right? But the answer is just, it's incredible. How does a tiny pill, once it's in your system, know how to hunt down and destroy invading bacteria while leaving your own cells totally unharmed? Well, that's exactly what we're going to break down right now.
So, to really get this, we first need to know our weapons and kind of survey the battlefield. Let's define the key players in this whole fight.
An antibiotic, at its heart, is a super specialized weapon for fighting bacterial infections. And what's really cool is that the first ones, like penicillin, were actually discovered in nature. They were compounds that organisms, like fungi, were already making to fight off their own bacterial enemies. We basically just borrowed a strategy from nature's own playbook.
Now these weapons, they don't all work the same way. They pretty much fall into two main camps. You've got your bactericidal ones. These go straight for the kill, just destroying the bacterial cells outright. Then you have bacteriostatic antibiotics, and they play a different game. They just stop the bacteria from growing and multiplying, which gives your own immune system the chance it needs to come in and clear them out.
Okay, and this is a really crucial point. Antibiotics are specialists. They are only designed to target bacteria. They have absolutely zero effect on viruses. That's why your doctor won't give them to you for a common cold or the flu. Using them when you don't need them just makes them less effective for when we actually do.
So how do they actually launch their attack? It's not just one single method. Nope. Antibiotics have a whole arsenal of strategies. We're talking 5 primary attack plans for taking down bacteria.
And these are the 5 core tactics. An antibiotic can act like a battering ram and just shatter the bacteria's protective outer wall. Or it can puncture the inner cell membrane, which causes fatal leaks. It can shut down its protein-making factories, stopping all construction. It can even scramble its genetic blueprint, its DNA and RNA, causing total chaos. Or it can block it from making essential nutrients, basically starving it to death.
Each one of those attack plans corresponds to a specific class of antibiotic. So whether it's inhibiting cell wall synthesis or disrupting the cell membrane, each drug is engineered to exploit a different, very precise vulnerability in the bacterial cell's machinery.
Now, of these 5 strategies, one of them really stands out as just brilliant. And it's the key to answering our original question. And that's the strategy of attacking the cell wall. Let's dig into why that is.
Okay, here it is. This is the answer. Our human cells, they have a flexible membrane, but we don't have a rigid outer wall. Bacterial cells, on the other hand, they're protected by this super strong wall made of something called peptidoglycan. It's like a unique mesh-like armor that only they have. And that difference, that's the secret. Antibiotics that go after the cell wall can attack bacteria with pinpoint accuracy because they're aiming for a structure our own cells just don't have. It's the perfect targeted strike.
But wait, the story doesn't end there. Attacking the wall kind of sounds like a brute force thing, right? But the reality is so much more sophisticated. We're about to unlock a whole deeper level of how these drugs really work.
Yeah, it's not about just smashing the wall down with a hammer. The mechanism is intricate, it's precise, and honestly, it's beautiful in its biological cleverness. The antibiotic acts less like a wrecking ball and way more like a saboteur.
So here are the true targets. See, antibiotics like penicillin, they don't just attack the wall itself. They target the workers, these specific enzymes called penicillin binding proteins, or PBPs. These little guys are responsible for building and maintaining that wall. So by disabling these essential workers, the whole structure gets compromised and it just collapses. And this brings us to the final and I think most fascinating part of this whole thing. If the PBPs are the targets, then you can think of the antibiotics as a set of master keys, where each one is designed to pick a different and very specific lock.
Just look at this incredible precision. Depending on which one of these PBPs an antibiotic targets, the result is completely different. So if you inhibit PBP1, which helps the cell get longer, the bacteria just bursts almost immediately. But if you target PBP2, which maintains the bacteria's rod shape, it turns into this weird round blob before it eventually dies. And get this, if you inhibit PBP3, which is needed for cell division, the bacteria can't separate, and they form these long, strange filaments.
And this right here is the whole point of modern antibiotic design. Different drugs, like the ones in the common beta-lactam family that includes penicillin, they're all engineered with different affinities to bind to these PBP locks.
So one antibiotic might be really good at picking the PBB3 lock, while another is a master at targeting PBP1. It's this highly specialized chemical game of locks and keys.
So the big takeaway here is that antibiotics are not the blunt instruments we might imagine them to be. They are incredibly sophisticated, highly specialized tools. They work by identifying unique features of bacteria and then targeting their precise molecular machinery with almost surgical accuracy. And this isn't just for the cell wall. Similar strategies are used to sabotage protein factories or scramble DNA, always targeting processes that are unique to bacteria.
But all of this leads us to a final and pretty urgent thought. This whole system of locks and keys, it's at the very heart of the huge challenge we face today: antibiotic resistance. The bacteria are constantly evolving. They're changing their locks to make our keys useless. So the question that's really going to define the future of medicine is, how are we going to design the next generation of master keys to stay one step